Okay. So, good morning. It's a Saturday and weekend, and you all had lots of talks, exciting talks during the week. I'm sure your brain is full of lots of ideas and stuff like that, and probably you're a little tired too. So I will try to say little and also simple. And if you have a question, it would actually help me if you ask in between. Just stop me and ask for any clarification, any question. Don't think it's a dumb question or anything. Just ask it, okay? It would help me. Okay, so I'm a statistical physicist by training, and much of my work these days is on evolution, specifically population genetics. So I'll explain the meaning of these words. And my job today is to show you how you can use ideas from statistical physics and theory of stochastic processes to understand or address questions in evolution. Okay. So let us start something from the very basic thing. What is evolution? So again, that's something you all know. You have read something in newspapers, in some popular science magazines and stuff like that. But let me say that for you anyway. So the point is that in a population, there is variation. So we all look different. Not just that, our DNA sequences are only a little bit different from each other. And some of this variation is heritable. That is, it can be passed on from one generation to another. And these variants, if they happen to be good in an environment, then they are selected for. That is, they will have probably a better chance of survival, maybe they'll have more offspring, and so on. Okay, so these are the three key characteristics, key things that you need for evolution. And in Darwin's words, therefore, natural selection is scrutinizing the slightest heritable variations, rejecting that which is bad, preserving and adding up all that is good. So that's basically the crux of what evolution is. Furthermore, he says that we see nothing of these slow changes in progress. We only see that the forms of life are now different from what they formerly were. That is, evolution you cannot watch. It's something we know that, chimps or something, and now we have something here now. And as you know, it's a very exciting time actually. Probably you read about this, that this year's Nobel Prize in Physiology and Medicine was given to a human geneticist. So to understand how the evolution works, how we are related, the modern humans, to the previous ones and so on, right? So this is a very exciting area. And then, however, Darwin says that, we cannot observe this in action. Well, we know several examples where we can observe evolution on some reasonable timescales. And I'll give you this example, which all of you have read in school. This is this classical story of the evolution of the black and white peppered moths. So this story is set in England during in the pre-industrial revolution era. These trees used to be covered with this object called lichen. Okay, it's algae plus fungus, some sort of symbiotic relationship between them. Okay, and that's the environment in the pre-industrial revolution era. And there is variation in this moth population. There are the black ones and the white ones. Okay. Then in this environment, however, the white ones are selected for because they are less visible to the predators. And they were the wild type, the common type. Their frequency was very high, like 90% and above. But there is this variant. It's not zero population. There's a smallish population of this. Then during the Industrial Revolution, the trees got covered because of pollution. Trees got covered by soot. And then it's the black ones, which are less visible to predator. That is, these variants are selected for. And then, again, in the cleanup in the last century, current trees in England are again covered with lichen, and it's the white ones which are selected for. So this is that when the environment is changing, how the different variants are selected for. Very important to understand that you need variation. Had there been only one type of moths, okay, then this moths population would simply have gone extinct. Okay. Other point which is important on this slide is what Darwin said, that you cannot observe evolution. But these large changes in the frequency of these moths, it happened over only a shortish time span, just about 50 years. Large changes from 0 to 1, roughly from 0 to roughly 1 and back to roughly 0 for the black pepper moth. So this is something that you have read in school. And there are some nice review articles which I strongly recommend if you would like to read up. Okay. Now, next one, I'll give you an example from a lab. So this is a long-term microbial experiment. It was started by Lenski in 1988, and this experiment is continuing to this day. So experiment is very simple. The idea is very simple. So what he has is just the usual E. coli, which lives a lot of them in our gut, okay? And then just take these usual ones and plate them in 12 plates or make a mixture of them. 12 such different populations, all founded from the same ancestor. However, you challenge them, you give them a new environment in which they are given less glucose than what they are used to. So now this initial population is maladapted. It's in a different environment, and if it wants to, if it survives, that means it has to accumulate good mutations which would increase its fitness, which would allow it to survive in this new environment. Okay, so that was the sort of idea to test, to see evolution in action. And because E. coli, it divides in about 20 minutes or so, unlike moths, we could potentially see a lot of stuff in a few years' time. So that was the motivation of starting such a long-term experiment. So this is the result, and this is almost a 10 years old data, in which we found that the average fitness, that is average over these 12 populations, as a function of time, this relative fitness, here fitness simply means the growth rate, okay? So that fitness increased a lot initially, almost 40% in the first few thousand generations, and it's still increasing away, okay? And much slowly, but still increasing. And these are the points from the experiment, and there are these green and red lines. They are coming from a theory. Now, experiments we can do, and the point here is that now people are very much interested in doing theoretical evolution. I mean, people have been doing it. It's not like suddenly people have started to do, but matching experiments and theory. So that is something which is now around, like, I think last 2-3 decades has gained a lot of momentum. Okay, so this is from the Lenski experiment. Yeah, please go ahead and ask. Yeah, so that means we need to look at the genetics of it, right? Yeah, so I'm going to show a very tiny part of that. Okay, so this is, so what I showed you in the previous slide was a fitness or a growth rate, which is sort of a macroscopic quantity. Now let's go to microscopic. So that's why she's asked us, Sandhya, what's happening at the microscopic level, at the genetic level. And now, because of genetic sequencing, we can tell you what is happening. Yeah, I'll answer that, because no, it's not flattening. No, it's actually, no, this is actually not a correct theory. Yeah, so the green one is the correct one. And its fitness is still increasing, but much slowly. Yeah. Okay. So now if you look at the microscopics, that means if you look at what are the changes happening at the genetic level, we can trace them, we can look at the dynamics of that. So this is a very tiny part. These are, 60, 70,000 generations, and I'm showing you just few 108,000 generations here. And there are, of course, 12 such things, colonies, and I'm showing you one colony. And I recommend to look at this paper if you want to understand it more. So what I'm trying to show here is that you see some mutant arose, and it swarmed the population. Its frequency became 1 in the population. The other ones, like this green one, it arose, and again, it spread to the population. But then there are lots of such mutants which again arose, but they did not make it. They arose, died, and so on. Okay, so there's a lot of stuff going on here. You just see this teeny stuff in the beginning, little increase in the fitness. But what's going on at the microscopic level? And you, as a statistical physicist, are well positioned to understand this point. This is like asking some question about thermodynamics. And now I'm asking that if you do this detailed microscopic modeling that we do in statistical physics, that's the sort of analog of genetics here. Okay, so those configurations, how are they changing with time and so on? Those are the kind of questions that one is interested in asking when we look at the genetics. Okay, So these are the dynamics in which lots of mutants arose, some spread, some not. So there's a lot of this stuff, and because of this, lots of mutations which became available initially, that gave rise to this large increase in fitness. But as the population adapts, it finds harder and harder to find better mutants, leading to a slower rise. Now, these two theories have some detailed differences, which I can speak about later on. Okay? Okay, so there are, this couple of examples that I gave you from ecology experiment and an evolution experiment in the lab. It poses lots of questions, and there are some questions that I am interested in, and I'll just tell you what. So, for example, one can ask questions about what are the dynamics of adaptation. That is, how do you understand these fitness curves? Not just in landscape experiment, but more generally. Will they always increase fast? What will be the slope like? Will they saturate? Will they just continue increasing? What should you put in? What kind of processes, dynamical processes you should put in to get those curves, to understand those curves? Okay. I spoke to you about fitness curves, which were these macroscopic things, or the color of the moth, which is a macroscopic thing. But there's some genetic one. There are dynamics going on at the genetic level. There's some dynamics at the phenotypic level, that is the color of the moth. If you look at its frequency, that is changing with time. How are these two things related? What's the connection between this macroscopic and microscopic dynamics? So that's another question. In the landscape experiment, the colony was starved once, and after that, it's always given the same amount of food. But if you think of the moth problem, pollution level is not a discrete variable, it remains the same. It's probably going to change, and fluctuate a lot over these 50 years or so. So if the population is experiencing a continually changing environment, how does that impact the dynamics of adaptation? So, and this is the last point, which I will say a little bit more. Okay, so this is a sort of very quick, I think this is not started. Okay, okay. So this is a very quick overview of what evolution is. Okay, but now I'm going to go into some details, especially quantitative details, which as a StatMech person, you should be able to appreciate and understand. So if there's any question, I'll address that first and then move on. Okay, everything is clear? Very good. Okay, so as we do in physics, we know some processes, some elementary processes we will take and then put them together and build a model. We know what are these elementary processes in evolution. Some of them you already know. One is selection. Here I've written natural selection. It can also be artificial selection. Selection simply means you select the good ones. The good ones have a higher probability of survival or more offspring, etc. Selection, however, has a tendency to reduce variation because, there's this pool and I select some of you, so that variation is gone. But variation is the driver of evolution. While selection reduces the variation, mutation creates variation in the population. And again, last 2-3 years of this pandemic, you have read in newspapers, this variant, that variant, and so on. They are very important. They are like pain in the neck for us, but that is what is driving the evolution of coronavirus. Okay? So that's one process, that is the mutation, which will generate these variations. Another process is called genetic recombination. Don't worry too much. All this means is that if I have some bunch of sequences, they can break and swap the parts so that you create new sequences, and that's how you can create variation in the population. So genetic recombination or sexual reproduction, they are other creators of variation. For today's talk, what is very going to be important is another evolutionary force called random genetic drift, or in other words, for us, stochasticity. This stochasticity is, however, agnostic about increase and decrease in variation. It can increase or decrease, it can do both, unlike the first two. There's yet another one called the gene flow, which I'll completely ignore today. So the point is that the population is structured. So there is some island in which some population is doing its thing, another island in which it's doing other thing. So there's structure in that, right? Today I'm going to assume a mean field assumption. That is, these populations are well mixed, or there's enough migration so that they are well-mixed populations. So I'm going to ignore spatial structure and other structures today. Okay, and the key point is going to be, because you're a statmic person, what is the role of stochasticity? That's the sort of the key point, the key message I want to get across today. Okay, so now that we have some broad categories of evolutionary processes, we want to know, because of the interplay of these various things, some create variation, some decrease variation, some are stochastic, some are deterministic, and so on. How does the frequency, and I'll introduce this word for you, allele frequency, change with time? Again, don't worry too much into new words. If you like, just think of it as upspin and downspin. It's just saying if I have a gene, it comes in two varieties, up and down, or black and white, something like that. That's it, okay? Okay, so we want to understand, under the action of these basic processes, how does a frequency change? What are the dynamics of it? So there are two broad approaches in evolution that one uses to understand, to address these questions. One is, again, if you are working in non-equilibrium StatMech, this is the approach you do, in which I give you some basic dynamical rules, and then you run your simulation or your calculations forward in time. In this case, what it means is I have these two alleles, red one and the black one. Maybe there's selection, maybe there's a mutation, maybe there's stochasticity, whatever. You put in all those details and run your program, and then at some point you will see some new frequencies, and you want to understand how that happened, what were the dynamics of this. So that's the kind of question that we address in population genetics theory. There is another approach, which is called coalescent theory, which says that, okay, you give me these samples today, and looking at this, there are this many reds, that many blacks, and so on. Can I say what were the processes which shaped this variation that I see today, the population structure I see today? Can I say that? Can I infer what were the processes and what were the magnitude of them? Okay, so I mostly work in this way of thinking that is going forward in time, and that's what I'm going to focus on. Okay, so let's ask a very simple question here, and we will take again sort of a physicist way of thinking. There are all these processes. I will turn off some of them and turn on others, and the way we think, I have 5 things, so I will switch off 4 of them, study one of them in detail, then turn off and turn on another one, and so on, and then hopefully arrive at some kind of complete understanding. Okay, so that's the kind of way I'm going to explain things to you today. Okay, so the question I'm asking is that suppose we have this population and a variant appears. How does this frequency of this new variant changes under the action of these various evolutionary forces? For simplicity, to start working, let us ignore stochasticity. Okay, it's a very difficult thing usually. I mean, I find it very difficult to handle stochastic things. Okay, I'm also going to assume that there are no further mutations. This mutant has come, and I'm going to work on those time scales where no new mutations arise. It's not that the theory is not complete. Of course, we can include mutations, but today I will not discuss that. Okay, so now we have only selection, and I want to know how does the average number of these mutants will increase with time. You are all experts in this. You have seen such equations before. So you expect that, okay, I started with one of them, one will become perhaps 2, 2 will become 4, and so on, right? If it's a good mutant, that means its number will increase exponentially. In other words, you will write dn by dt equal to s times n if s is positive. That's a growth rate, exponential growth rate, right? But of course, population cannot keep on increasing forever, right? So that means there must be a finite carrying capacity. It should saturate at some point, and that's why we have this other term. Okay, this is the standard equation which I'm sure you have seen in other contexts called the logistic equation. Okay, so in the stationary state, which means you put dn by dt equal to 0, you have 2 zeros. That means n can be either 0 or it could be capital N, which is the carrying capacity of the population. So what do we expect naively? If you asked me, I would have said, okay, because if this mutant is a good mutant, it's a beneficial mutant, its number would rise and its number would become capital N. But if it's a deleterious mutant, that's a bad mutant, or then in that case, its number will decrease to 0. Right? That's our naive expectation. I want you to keep this in mind because our answers are going to change. Okay, so but this is the prediction from, yeah? What is it? Yeah, thanks. This is the fitness of the mutant S relative to the wild type. Yeah, here I'm considering not frequency dependent, yeah. But that's again more generally one can do, yeah. We just have two types. Just two types. The black moth and the white moth, that's it. Okay? Because, again, not because it's not doable, the kind of questions you're asking, because it's a Saturday, and therefore we will not to discuss that. But I'm happy to discuss that later on. Yeah? Let's just do very, very simple things so that we know how to go to the more complex things. That's my attitude. Capital N is the total number of individuals, yeah. Any other question? Okay, good. Okay, so this is what we think, right? I mean, okay. But this is a deterministic theory. Again, perhaps you also have heard of it as rate equations, right? These, of course, neglect stochastic fluctuations, and stochastic fluctuations are very important when numbers are small. Okay? So when a variant, just new variant, our new Omicron variant just appeared, don't forget, we are seeing that it has swamped through the global population. But think of it when it just appeared. It was small in number. At that time, it's not correct to write such an equation. Okay? So when it's small, at that time, it's very important to take stochastic fluctuations into account. So that's what I'm trying to now convey to you. So let me explain that using a very simple cartoon model of stochastic models. Again, let's right now, for simplicity, ignore selection and no further mutations. And I'm going to consider just this silly small cartoon in which I have 6 individuals. Total capital N is 6. And these are the squares and circles. Squares and circles, they are equally fit. They are not better or worse than each other. And there is, of course, genetic variation because, this is square and that's circle. Think of it like maybe a bacteria. It divides so that instead of 6, I got 12. But there's food for 6. So half the population has to die. Right? But they're equally fit. There's no reason why this is more likely to die than that one, right? So what I can do is to toss a fair coin and kill half of the population. Right? Okay? I can do that. I tossed a coin and I got this. And then I found that, yeah, instead of 3, 3, I got 2, 4. Right? So that's how, by random sampling, stochasticity is entering into the picture. There are other ways of taking into account, but this is the one I'm going to focus on today. Okay? Right? So this is how we are going to take care of stochasticity. Let's go a little bit more, one more generation. I had 3 squares, 3 circles. I removed the intermediate steps, but it has so happened, as I said, 2 squares and 4 circles. In the next generation, these will produce 4 offspring, these will produce 8 offspring. But because of heredity, you copy your parents' genetic material and I'm ignoring any mutations, then because there are more of circles, there's a higher chance than in the second generation, there will be more circles. Okay? That is the effect of heredity. So this is all verbal. We can now think of this stochastic process, write down a Markov chain for it. So let me do that. By the way, I noticed that I think in the first 3 days or something, you had lectures on stochastic theory and first passage and Fokker-Planck. So I'm assuming all of you are experts. Okay? So this you should be able to understand. Okay. So therefore, if you're thinking of this finite Markov chain, then the transition probability, that is, if I had I squares in generation G, then in the next generation they became J, that is binomially distributed, but the success probability depends on the previous generation. So this is not your usual coin tossing problem in which from one toss to another, the coin remains fair. I started with a fair coin, you see, equal, equal, but because of this heredity, it became a little unfair. Unfair meaning the probability now depends on the previous generation. Okay, so this is different from the simple textbook problems in stochastic processes that you might have read in your coursework or in your research. This, we can using this, we can either work with the Markov chain, or you can write down a Fokker-Planck equation for this. If you do that, it works out, quite easily, that the equation is not dp by dt equal to d2p by dx square. You get this additional factor. And that origin of that is over here. Okay? So this is a different equation. The solution is not a Gaussian function. It's in fact called Gauss hyper. It's a special case of what is called a Gauss hypergeometric function. This problem, it turns out, is a exactly solvable Fokker-Planck equation. Okay, so now we are getting into new territory. Not quite new. These equations you know, but they are different models, where this term came because of heredity. Let's continue. So 3 we started, maybe it became 2, maybe it became 1, because it's a, it's fluctuation. So it's not like 1 will just go away. Maybe it became 2 and so on. But I guarantee that if you run this program long enough, you will wind up in a configuration in which all are squares or all are circles. Once you get into squares, suppose you get into squares, there's no way you can get out because I'm not allowing any further mutations, remember? Okay? So these are called absorbing states. These are an example of non-equilibrium stationary states. And there's a lot of work and techniques and so on that people try to address. And I think you will hear more from other physicists in the next week about them. Okay, so in this example, it happened to be that all became squares, but of course, if you run it again, all can become circles. Now, when you see this for the first time, it's not very clear. How can all become squares and circles? They were equally fit, remember? And I started with the best initial condition, equal number of them and equal fitness. Yet, one of the population took over. So that happens because of its rare stochastic fluctuations. Okay? So now because we have two absorbing states, okay, we can ask, So this was, so far I assumed that they had equal fitness. We can easily generalize it and put in a model in which they can have different fitness, okay, and run the same kind of program. And suppose this black mutant had higher fitness, maybe it spreads eventually, or maybe it doesn't, okay. So it's not something special just about equal fitness. Even with selection, you will hit these absorbing states. So this brings me to this, what is called the first passage problems. So these are the stochastic trajectories in which, starting from the same initial condition and with some selection, some trajectories, this mutant became 0, in some other trajectories, it took over the population. And the typical questions that one asks in this business is, what's the chance of reaching an absorbing state? That means what's the chance that the population spreads or it gets lost? In physics literature, it's called splitting probability. In population genetics literature, it's called a fixation probability. And another classical question that one can ask is, how much time does it take for the population to, let's say, spread? And that is called a first passage times. Okay. So these are these classical questions that one can ask here. And there's a standard techniques that one can use to address them. Like already you heard in the first lectures in this week, we can write down this backward Fokker-Planck equation, which says the probability that the mutant frequency is x at some time t, given it was some x naught at the previous time. And for this process, it's called Wright-Fisher process. In the scaling limit, where n is large and the selection parameter is small, such that these product is finite, in that case, you can derive from some basic Markov chains this kind of Fokker-Planck equations. So this is the term which I showed you already because of stochasticity, and this is the deterministic term coming from the logistic equation. Okay, so this is the most general Fokker-Planck equation that you can write down, at least, most general in the sense that I have this derivative is still there. And you can work out things from it. So just to give you an answer, if I ask what's the probability that the mutant's frequency is eventually 1, then starting with some initial frequency, setting this equal to 0, you can find this result. And to students, I would recommend to try and solving this and try to understand the properties of this. For today, I will just mention the following qualitatively different result from what we discussed at the beginning of this lecture. Remember what we said? If I have a beneficial mutant, yeah, it will spread, it's a good guy. No, if you take the stochasticity into account, what this says is the chance that the beneficial mutation can get lost is also non-zero. Yeah. No, it's a Markov chain. But the question is not Markovian. The process is Markovian, but the question is not. Question is asking, you have not hit that 0 or 1. Only one generation. Yeah. So, yeah, just the last generation. So it's still a Markov chain. Okay? Yeah. Any technical questions? Okay, very good. Okay, so the simple thing I want you to keep in mind is that just because you're good does not mean you have a great paper, innovation. I mean, I'm making it sort of, more social stuff, but maybe it helps you to remember, is that if it's good, it does not necessarily mean that you will win because of stochasticity. Just because you're bad does not mean you'll lose. Because of stochasticity, it's possible that a bad variant can spread. There's a small chance, but there's a non-zero chance. Okay, so this is a very simple qualitative result. It is difference which we see compared to the simple deterministic theories that we wrote down at the beginning of this talk. Okay, yeah. So that's all fine. I'm a statement person, and most of you are also, and good. We can calculate lots of first patches stuff, and happy. But why would our biologists care about this first passage thing? Why should Sandhya listen to my talk? Right? So I would like to convince her to listen to my talk, and therefore I need to make connection with biology. I will try to now show you that these little quantities that we spend a lot of time as a physicist, they actually can be related to the things that biologists can measure or measure routinely. Okay, so to do that, I need to take you more towards evolution side and introduce to you a phenomenon called genetic hitchhiking. Almost all of you probably are hearing it for the first time, so I will go a little slowly. So I'm thinking of, let's say, 4 individuals, and they have this long sequence. I'm showing you only a part of sequence. So some are circles, some are squares. So this is the sequence of a single individual. This is a sequence of second individual, third, and fourth. Okay, so I have this population of 4 guys. Okay, so there is variation, right? This sequence is different from this and so on, right? Let's say in this 4th individual, some mutation arises, right? And if it does not get lost, because remember, mutations can get lost. Whether they are good or bad, they can get lost. Provided it does not get lost, it will spread to the population in some time, which is our friend called the first passage time, right? This is true if I have an asexual population. There's nothing really which is completely asexual. Usually there is some amount of recombination sex, which just simply means that these sequences, they can break and once in a while swap the material. And that happens also on some time scale, for example, the inverse of the recombination rate. Okay? So the point is this. If I have this sequence which are physically linked, these variants which are physically linked, and I don't break these sequences, then while this is spreading to the population, the variant which is physically linked to it will also hitchhike with it. Okay? So this has hitched a ride with this guy and spread it now to the population, but now what we have got is identical sequences. We have lost genetic variation. Of course, if they could swap and break and so on, but not too much, then we will see more or less still different sequences. Okay? Now this loss of genetic variation is what biologists can measure. They routinely measure these things. So, for example, if you have sequences, all you do is a measure is to just calculate the average Hamming distance. That means wherever they are unequal, you count one. Okay? So that average Hamming distance is a measure of the genetic diversity in a population. So that's done all the time. And if you plot that as a function of your sequence, so you have some sequence, then the pattern of these genetic diversity, if hitchhiking has happened, is of this V-shaped pattern. In the position where the selection was operating, the genetic diversity is very low, and far from it, it is the usual standard one, I mean, there's some number 1. Okay? So these patterns can be generated, they are measured by biologists, and this is used to find the sites which are under selection, and the depth of it is a measure of the magnitude of selection. So this is done, and because, I mean, I know from a certain formula which I have not shown you, from this formula I know how much will be this depth, and what is entering in that formula is the first passage time. Okay, so that is the connection between what biologists can measure and what I can calculate. So this is an example of that kind of connection. Okay, okay. So this is doable, and there's a lot of work. So if you just search genetic hitchhiking, you will get tons of papers. Okay, this is something which people have been thinking for almost 50 years for various reasons. Okay, okay. There's a problem with this thing that I just described to you. Theory predicts that you will see same depth, whether this mutant was a good mutant or a bad mutant, you will see the same level of reduction. Okay? And that's because these theories assume that the environment was not, it remained constant over the time scale when hitchhiking was happening. That result comes from a property of these conditional first passage times. I'm not showing you the equation, but it turns out is a remarkable symmetry. The conditional fixation time for a mutant who has a advantage plus S is same as that for minus S. And because these first passage times are equal, therefore the reduction in diversity is also same. Therefore, we cannot distinguish whether this reduction is due to a beneficial mutant or a deleterious mutant. So our contribution is to say that, you're assuming that the environment remained constant, like in Lenski experiment. If you allow environment to change, we find by the analysis of a Fokker-Planck equation that this symmetry does not hold, which means you will see different levels of reduction, and that could help one to distinguish between positive and negative selection. So let me just elaborate in like 5 minutes on that last point. So this is the work done with my former PhD student, Sachin Kaushik, who is now a postdoc at Cornell. Okay, so, we should not, of course, in natural environments, they do change all the time, right? It's not like just change once and just stay put, right? They change continually, right? And so therefore, we were interested in understanding how does this continually changing environment impacts various objects of interest. So I'm thinking of seasonal changes. So for example, a mutant, maybe it's favorable in summer. It tries to spread and increase frequency. But winter came where it happened to be unfavorable. So its frequency decreased. If it escapes stochastic loss, then maybe it will spread, right? So you have these environments which are changing, and you're asking questions about what's the first passage time in these changing environments. We can write down, of course, our Fokker-Planck equation. The machinery is there, and we can exploit that. So the equation now we have is that this coefficient, for me, selection is now time dependent. This is technically slightly challenging problem, because if you had a constant environment, you have time translation, you can get rid of, for station, if you want to know things at last times, you can put this left-hand side to be 0, but now you cannot. Your answers would, because of inhomogeneity in time, answers would depend upon initial state also. So it's a slightly more complex problem. and you have to deal with the PDE, partial differential equation, whereas if you have a constant environment, you can work with the ordinary differential equation. So it's technically slightly challenging. Moreover, we have 3 time scales now. One is with selection occurs, and time scale over which stochasticity happens. That was there in constant case also. But now we have additional time scale, that is the one in which environmental variation occurs. So we could use standard methods like perturbation theory to get these expressions for mean first passage times when we have a slow environment or fast. Slow and fast, I simply mean that the environmental rate is the smallest or the largest parameter. So in that case, we could obtain some analytical results, but I'll not show that to you. Okay, fine. So we can do all the math and everything and come back. What we learned is that the conditional first passage time is indeed different for plus S and minus S mutant, and because this first passage time relates to genetic variation, if I measure that in the simulation, then we found indeed that this is the genetic variation in the beneficial case, and for the deleterious it's different. If there was no environmental change, these 2 points will be coinciding. Okay, so that was the whole point of this exercise. And what good is it for? I mean, so what we are suggesting to our evolutionary biologist friends is that, if you assume constant selection, then of course you cannot find the sign of selection. Not only that, because if environment is changing slowly, you might wind up inferring incorrect parameters because you're inferring it from the depth that V valley, the depth of that V valley. Now, if environment is actually changing, even slowly, this depth may be higher or lower, and therefore your inference of the parameters would be wrong. So we suggest, therefore, to include in your inference problems, to include the possibility that environment might be changing. So that is sort of the our suggestion. Okay, we can do more general things, not just Fokker-Planck equation. Why stop here? We can always also include demography. That means population size need not be constant. It could also be changing in time, and that is very important. Out of Africa, when we came out, there's a population bottleneck. Population does not remain constant in time. Okay, so but we can work out all these things, which I will not show you today. But basically, the simple result which emerges from Sachin's thesis is that genetic diversity is qualitatively different from that in static environment, even if environment is changing slowly. That is, even though these parameters change slowly, the genetic diversity is different. Okay, so I think I can stop here. I said a lot of stuff. Some of it might be very new to you. Some of it you could recognize, and if you're interested, you can have a look at it. But these are the simple lessons that I would like you to take home. First of all, evolution is observable. Okay, and I showed you a couple of examples, but there are lots and lots of very well documented examples of evolution in action. When we read newspapers, we read these science magazines and so on. Everybody talks about selection, mutation, and so on. What they don't tell you is this stochasticity. So next time when you read about new, coronavirus variant or something, remember, you're able to see it because it escaped stochastic laws. Don't go just by the fact that a mutation happened and the variant came. Oh, it's in a good environment, it's selected for. Yeah, but it also got lucky. Okay, so as a StatMath person, you should be able to appreciate that very well. So this is the key lesson that I want you to take home. Evolution is a largely stochastic process, and the stochasticity can come in various shapes. I focused on one type, okay, but mutations I ignored completely today. Environment could be changing randomly. I assumed deterministic environment changing, but it could also be. And it's a very nice playground for statistical physicists, and that's why I'm interested in it. We talked about first passage properties, but we can have interesting stationary states which are not in equilibrium. So we could study all that. So with that, I think I'll stop here. Thank you. Yeah.
Okay, I have a question. So you said in presence of static environment, the good mutation or bad mutation, it does not distinguish. It just have to be lucky, say, one survive over the other. On the other hand, when you are using...
No, so the first statement is not what I meant. or maybe I didn't say it clearly. The diversity produced by this phenomenon of genetic hitchhiking, that pattern, that level turns out to be not dependent. But whether or not you survive, that does depend upon the selection.
Okay.
So the
depend.
beneficial mutation, as your intuition suggests, beneficial mutation should have a high probability of survival. Right? That does happen. That's the Kimura's formula. And a bad one should have a low probability, but non-zero. That does happen. But the patterns that are produced, at least by that phenomenon, they unfortunately are not able to distinguish if we assume static environment.
And and for a changing environment, how does that?
Yeah, so that changed, the numbers change. So I showed you only a tiny part. So these numbers would have coincided, but now what I'm showing here is the rate at which environment is changing. With that, so these numbers would have been coincident if I put x-axis equal to 0. But now they are different. It's like splitting of the two levels. So they are different numbers, yeah, and substantially different numbers. Yeah. It depends on initial conditions because it's inhomogeneous in time. So this example, it happened to, yeah, it can decrease also. Yeah. Yeah. So which time scales would you like to distinguish, this one and this one? Yes? Yeah, I'm assuming they are very different. So environment is changing slowly and selection is very slowly compared to the time scale over which selection is acting. So, I'm just saying that you can fill in the whole thing. You can, have all the parameters of the same scale, very different and so on. You can put in the full picture, but that you have to do numerically. Analytically, we could do only some limiting cases. I think there's a question behind you. Yeah.
Just following up on this last plot that you showed, I mean, you just said that this difference is conditional on the initial conditions. But in a real experiment, the mutation will arise at some random time point. So effectively, you would have to average over
Correct.
the times
That's
where
right.
that. Then don't you symmetrize it again? Don't you get back to what you had before?
That is correct. So therefore, what I would now take from this is not so much that this is different from this, but the fact that this is different from what if environment was constant. So therefore, what you're saying is right. I should average it over the initial conditions, right? And therefore, they will collapse together, right? That's correct. So yeah, it can be different if I knew what the initial condition is, but I think the main takeaway then is that this depth would be different if you allow for changing environment. That's all. Depths are different, so don't distinguish between beneficial and deleterious necessarily. Now, distinguish within beneficial when you have a constant environment and a changing environment. So that is how I would interpret it now. Yeah. Yeah. Is preferable for the species to yeah. 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 No. Okay, so I think I was not clear. Only this particular thing, which is the pattern due to hitchhiking phenomenon, that does not distinguish. Other properties are different and gel with your intuition. Okay? Yeah. Yeah, please. Yeah. Yeah, so that happens, of course. And if I'm thinking of a sinusoidally changing one, today is beneficial, in the winter it's unfavorable, right? So, you think of things like average over period, for example. and see if that makes a difference. So if the frequency is very high, it sees the average. So it's like it's just seeing the average environment. So that's one thing that you can do to quantify this. But yes, that of course happens. And therefore it matters where you started. And one reason I was focusing on slowly changing is simply because I wanted to not discuss this point about changing your identity. So you are today beneficial, tomorrow you could be deleterious. Yeah. Lunchtime. Thank you very much.